Thank you all for joining us today for our Distinguished Speaker Series. I'm Nancy Walters. I'm the Executive Director of the La Jolla Community Center, and I'm glad that you're here today. If you're not familiar with the Community Center, we are a nonprofit organization, and we provide programs and services that promote lifelong learning, wellness, and friendship. All of our programs are geared towards seniors, but they're open to all. Of course, during the pandemic, we have transitioned over to virtual programming, and we have a lot of offerings every day, everything from Distinguished Speaker Series, cooking classes, meditation, yoga, Tai Chi, all sorts of really fun programs. So I hope that you can not only join us today, but continue to visit our website for upcoming programs. So thank you again for being here today. Before we get going, I do want to give a big thank you to Judy White, who is the generous underwriter of our Distinguished Speaker Series. So this would not be possible without her. So thank you, Judy. And if anyone is interested in sponsoring any future programming, please just let us know. Give me a call or send me an email. All of our contact information is on our website, and that's ljcommunitycenter.org. So before we get started, I want to let you know a little bit of housekeeping. If you have any questions during the presentation, please feel free to use the questions and answers or the chat function at the bottom of your screen. That's going to be available, and I'll be monitoring those. And we will address any questions at the end of the presentation. So we're very excited to have Dr. Mobley here today. As we all know, we're all in the middle of a pandemic, but of course, that's not the only pandemic or epidemic going on. COVID-19 is not the only one. There's also the epidemic of Alzheimer's disease. And we're going to have Dr. Mobley tell us about where we stand in the fight against dementia. So we're really excited to have him here to let us know a little bit more information about such an important topic that we all face. So before we get going, I'm going to tell you a little bit about Dr. Mobley. Dr. William Mobley is an Associate Dean of Neurosciences Initiatives, Distinguished Professor of Neurosciences, and Interim Director of the Sanford Institute of Empathy and Compassion at UC San Diego. He also serves as Executive Director of UCSD's Down Syndrome Center for Research and Treatment, and holds the Florence Ryford Chair of Alzheimer's Disease Research. Dr. Mobley's research is focused on degenerative diseases of the central nervous system, especially Down syndrome and Alzheimer's disease. He and his colleagues have for many years explored the hypothesis that deficient axonal transport of neurotrophic factor signals contributes to neurodegeneration. The accolades and achievements for Dr. Mobley go on, and if we were to read them all, we would be here for a long time. So without further ado, I'm going to go ahead and introduce Dr. Mobley.
Well, thanks very much, Nancy. Really appreciate the opportunity to come and address your listeners. I want to mention that I'm a neighbor. I live in La Jolla, and I'm sure that I see you around from time to time, and certainly hope you're doing well today and that this season is treating you well. We're dealing with a lot of stress, and it's the stress of COVID-19, but it's also many other stressors. And highlight today is that what I'm going to say, I intend to be a little good news and a way forward that might help us deal not only with COVID-19, but with Alzheimer's disease. So with that, I'm going to go to my slides and I'm going to share my screen. And so hopefully you'll see my first slide, Alzheimer's disease, biology of memory loss. And again, as I suggested, maybe some good news around what we can do even now to deal with this disorder that affects so many. So my disclosures are just a few, but none of those that you see, except for those printed in kind of a yellowish color, are relevant. I am very involved in research on Alzheimer's disease, and I am a co-inventor on a couple of patents, but we won't talk about those today. I mean, we'll simply reference Alzheimer's disease in general and won't be speaking about any work that we're directly involved in that could have a financial implication. So what about Alzheimer's disease? And I define it as an oncoming epidemic. It's a common age-related disorder. It's characterized by the very subtle onset of memory impairment, which is followed in time by progressive compromise of cognition, ability to do the activities of daily living, and eventually really a disintegration of the personality and death. There's no cure for Alzheimer's disease at this time. There's no way right now even to interrupt it. So it's a very serious disorder. And it's common. Right now in the US, we estimate that there are 6 million people affected. Because of the aging of the population, more than 12 million may be with us in 2040. And the yearly cost at that time will be greater than $1 trillion, which is just a staggering sum, but quite understandable given the cost of care. Currently in the world, there may be about 60 million people with Alzheimer's disease. So a very serious epidemic with very serious consequences both for the individual and for society. Well, how do we prevent the epidemic? And there's a lot we have to do. We need to understand the underlying biology better. We need to discover treatments that really target the underlying mechanisms. We need to translate those discoveries to clinical application and test prominent treatments in rigorous, well-powered clinical trials. And finally, we need to understand when and how to best diagnose it and do it early so that we can get in there and help the brain out before there's significant dysfunction. So there are a number of insights that come into Alzheimer's disease. They come from a variety of sources. We can look at the brain and we can see that it's smaller, it's shrunken. Excuse me. We can look at markers at a microscopic level that are very characteristic of the disease. These are plaques and tangles and changes in endosomes, and I'll show you pictures of those in a little bit. There's also changes at the microscopic level in structure and function, and we'll talk about the connections between neurons called synapses, and they're lost. And we'll talk about the loss of neurons themselves, and we'll talk about reduced brain metabolic activity. And then we'll briefly talk about the toxic features of the brain, these molecular accumulations that are almost certainly injuring neurons and killing them. And then we'll also briefly talk about genetic factors that are very important for understanding all the things that make up the biology of Alzheimer's disease. So here is a very vital part of the brain. It's a connection of two neurons at a place called the synapse. And I'll just outline that word for you, S-Y-N-A-P-S-E. This is the place right here where one neuron talks to another neuron, and it does it as a result of an electrical impulse that comes down the axon and releases this neurotransmitter, these little gray dots, and they bind to receptor molecules here on this neuron, and they excite that neuron or inhibit that neuron. But the net effect is that neuron 1 here is communicating with neuron 2 here. And that's the fundamental basis of information transfer in brain. Now, a little more neurobiology. So what we've just outlined is this place here called the synapse. But of course, the synapse is part of a larger cell called the neuron. The cell body is shown here with its nucleus. It has these little spiky projections called dendrites. This is where it receives information from the neuron just in front of it, just upstream of it. And the impulses that are induced travel down this thing called the axon, all the way down to the synapse where they release the neurotransmitter that we've just looked at before. So this is anterograde flow of information. There's also retrograde flow of information from this neuron to this neuron. That information flows down the axon in the backwards direction. It's called retrograde transport. And these two flows of information, the to and the fro, are terribly important for keeping synapses healthy and functional. And one of the very interesting ways in which this neuron talks to this neuron at the synapse is through the formation of these things we call endosomes, signaling endosomes, that carry trophic signals released by this neuron to this neuron synapse at the synapse, and then those are internalized. And those little signals are carried all the way down the axon to this cell body. So the message here is that there's to and fro communication of information that's vital for keeping the nervous system healthy. And so here again, the synapse illustrating the to and the fro flow of information, and really understanding how critical that is for neurons to basically talk to each other. Now we know that when things are not normal, the brain changes. So here we're looking on the left side at half of the brain of a healthy brain, and on the right side of a brain affected, half of the brain affected by Alzheimer's disease. And what I want to point out to you is, notice this rim, this brownish rim, that's the cortex, that's the surface of the brain. And you can see it nicely wound around the surface all the way around the brain. And it's quite a rich, full brain where there's very little space between these two different folds in the brain. But notice how different it is on the right side, where that brown layer is thinner and where the spaces are much larger. And notice down here, this is a structure on the left side. This is the hippocampus. This is where early memories are recorded. And notice over here how shrunken that structure is. Very characteristic of Alzheimer's disease, and the reason, and the morphological reason, that people have so much trouble with short-term memory. And the overall brain then is quite atrophic. Notice that this space has become enlarged here because of tissue loss, and this space is also enlarged because of tissue loss. So the brain is shrunken. When we look under the microscope, we can see there are also changes that are very characteristic of Alzheimer's disease. So here we're looking at that cortex, and notice with the silver stain, one sees a lot of little sort of dark dots. And when we look at higher power, those dark dots consist of little collections of processes, both dendrites and axons, around a central core of amyloid. And the peptide that's present there is called A beta. This is an amyloid plaque. I'll come back to A beta in a minute. Near that plaque, one sees, for example, these very darkly stained neuron cell bodies, these filaments that are quite dark. And that protein that causes that is something called tau, or phosphorylated, or p-tau. And that's the tangle. That's the neurofibrillary tangle. And then a third characteristic feature of Alzheimer's disease pathology, I hope you can see it, are the enlargement of endosomes. So notice these little tiny black dots that you can see very clearly in A and C and B. And those are quite enlarged. And that's quite abnormal. And in fact, this change, the enlarged endosome, is something one sees before tangles and before plaques. So three cardinal features of Alzheimer's disease pathology at the microscopic level. Remember, if you will, that endosomes carry those signals that I've talked about. So this was our clue that those endosomes that carry those signals might be quite abnormal in Alzheimer's disease, and also other investigators felt the same way. Now, what about A beta? Briefly, A beta is a small peptide, so it comes from this larger peptide called APP, amyloid precursor protein. And through a series of enzymatic reactions, the brain produces this A beta peptide. And here I've called it A beta 42. So this peptide is in plaques, and it's in endosomes. And also it forms toxic complexes that injure neurons. Now I've mentioned tau, and I mentioned phospho-tau. Normally tau is very involved in keeping those railroad tracks that allow for that to and from movement to occur. But in Alzheimer's disease, one sees it falling off the microtubules and being abnormally phosphorylated. So tau is converted to p-tau. P-tau is found in tangles, and it's also found in soluble complexes that are toxic. So information flow in the brain is is abnormal and the synapse is under attack. P-tau is frustrating this to and fro movement at the level of the axon. A-beta is frustrating the transmission of signals from one neuron to another. And there's a traffic jam of these endosomes that prevents this trophic signaling to go back to the cell body to keep it well and healthy. And the net effect of that is there's a short circuit. This neuron used to be connected to this neuron. It's no longer connected. There's a short circuit. And because there's a short circuit, that circuit function is lost. And one of the manifestations of circuit function loss is, for example, inability to remember, inability to properly think abstract thoughts, inability to perform activities of daily living. All the things one sees in Alzheimer's disease are really the problems of short-circuiting synapses. When we think about the clinic and we think about diagnosing and treating Alzheimer's disease, we have good markers now, not perfect, but good markers to look at the extent of brain atrophy. And so here, a cartoon comparing a normal to an Alzheimer's disease brain, we can see these changes at the level of the MRI. We can see shrinkage at the whole brain, shrinkage regionally. And we can even see differences in the connectivity of those axons using a different tool under the control of the MRI machine. And we can also look at function. We can look under something called functional MRI or fMRI. We can look at the activation of brain circuits. And using a different tool, we can even look at glucose use. And that's decreased, as is activation of brain regions in the case of Alzheimer's disease. We can also do biochemical studies. So in spinal fluid, here shown CSF, we see a decrease in A-beta 42. Why? Because that peptide is accumulating in plaques. And we can see an increase in tau and phospho-tau. Using a different tool called a PET scan, we can show amyloid being deposited and tau being deposited. And finally, we can take blood, and we can see that in blood as well, there's a decrease in A-beta 42, an increase in p-tau, an increase in a protein called neurofilament light protein, which marks the destruction of axons. So let me give you some real examples of what we see. So here on MRI scanning, here the normal brain on the left, and here the Alzheimer's disease brain on the right. And notice, as I pointed out before, a nice thick cortex here, much thinner here, nice coming together of these different folds of the brain here. Notice space between the folds of the brain here. Nice small spaces called the ventricular system here. Notice how they're much enlarged here. And strikingly, a normal hippocampus here shown in a kind of a greenish color, and virtual absence, not complete, but almost the absence of that structure in Alzheimer's disease. So those are the MRI scans. And as I mentioned, one can do imaging to detect amyloid. So here we have a series of scans from a series of patients going up from a control here through an intermediate stage called minimal cognitive impairment to a stage called Alzheimer's disease. And what you're seeing is an increasing amount of red signal. And that red signal specifies the presence in brain of amyloid plaques. So these arise, interestingly, years before people develop cognitive problems. And about 30% of people over age 80 have positive scans. Doesn't mean they're going to have Alzheimer's disease, but they are already depositing amyloid in their brains. What about PET imaging? What about those tangles in that p-tau? Well, notice here we're looking at controls and minimal cognitive impairment and Alzheimer's disease here. And notice the red signal, very little, there's really no red signal here. This is a normal brain, no accumulation of tau. But notice as we progress, an increase in that red signal, first medially and then spreading over the cortex, here in MCI and here in AD. So we can image amyloid and we can image tau. And we can image glucose utilization. So here the red signal is good. Here's a normal brain. The red signal indicates that the cortex is metabolizing glucose. Notice the decreases that one sees in the red signal in mild cognitive impairment and here in Alzheimer's disease. So one now has great tools clinically to try to make a diagnosis of Alzheimer's disease. Now, genes matter. And so here I'm showing you, if you will, the genetic landscape of Alzheimer's disease. Some changes in the genome are relatively common. So in our call, 30, 40, even 50% of people might have changes in these genes called out here, for example, PCALM, and here BIN1. These are changes that are very common, and they have very little effect on the risk for Alzheimer's disease. So we're plotting risk here, and we're plotting frequency in the population here. So very common and not particularly important or particularly powerful in causing an increase in risk. Some factors that are more common do cause an increase in risk. So ApoE4, you probably have heard of ApoE4, causes increased risk. About 14% of the population, a smaller fraction, only 3 or 4%, and even higher risk of Alzheimer's disease. And then finally, genes that are known to actually cause Alzheimer's disease. So mutations in presenilin 1 and presenilin 2 and an APP. And here I point out the fact that in Down syndrome, virtually everybody develops dementia by the age of 60, and they have an extra copy of that gene for APP. You'll remember APP is the source of that A beta peptide. So there is a genetic landscape. Most of us avoid these very powerful changes, but we're certainly confronting a relatively low amount of risk from such factors as these. And notice, if you will, the color coding here, red showing a change in APP metabolism, yellow in tau metabolism, and here green in endocytosis. So the factors that we're looking at pathologically are recapitulated to some extent in what we're seeing in terms of genetic risk. Well, what about trials? And the truth is, we do not have disease-modifying treatments for Alzheimer's disease. This figure comes from Jeff Cummings and colleagues, published in 2018. And there are 3 categories of drugs that are being tested. Some drugs are meant to reduce symptoms. And these are typically small molecules, and many are being looked at so far. As I'm aware, none have proven to be effective. A second category of small molecules are small molecules that are meant to be disease modifying. And one can sort of figure out what their target is by looking at the color coding. So, excuse me, so here amyloid related in red, tau related in blue, and other approaches in gray. And notice that a number of drugs are being tested. And we're hopeful for these drugs, but to date, none has been shown to be both effective and safe. And then finally, you'll have heard a lot about immunotherapies that are meant to be disease modifying. Again, nothing has gotten us all the way to phase 3 plus with efficacy and safety. There will be data soon on a drug called aducanumab from Biogen, and it's possible that it will be approved, but there's no guarantee of that. And in fact, a number of scientists would argue against that because the data, at least in the public domain, is not convincing that aducanumab will be effective in causing an improvement or a lessening of the decline of people who have Alzheimer's disease. So no answers. And why are there no answers? And there's lots of reasons. The sources for failure have to admit to the fact that the disease that I'm talking about is quite complicated. We don't really understand all the mechanisms. In fact, we're looking at those mechanisms actively on a daily basis in our lab and in labs across the world right now. Not only do mechanisms exist, they interact and may reinforce one another. So for example, the fact that one has A beta in the brain may engage, and almost certainly does engage, inflammatory responses that could themselves be damaging. And all this is occurring in the context of the aging brain, which certainly is undergoing changes that may make disease biology more complicated. And remarkably, if one over the age of 80 who has a diagnosis of Alzheimer's disease comes to autopsy, one understands that only a relatively small percentage of those people have only the pathology of Alzheimer's disease, other pathologies then being present, including pathologies that are linked to other disorders like Parkinson's disease. And finally, there's a very real concern that interventions may be coming too late to stop progression. And so here's a curve from Cliff Jack and their colleagues that are looking at the degree of abnormality in certain findings, biomarkers, or clinical function, and disease stage here on this axis. So here we're going from normal to cognitively still normal to MCI to dementia. And notice that well before MCI and well before dementia, there's a change in A beta deposition and a decrease in A beta 42 in plasma and CSF. And tau-mediated injury is occurring well before one diagnoses memory loss. And in fact, brain structure is also changing before one gets to the level of memory loss and dementia. And here is the memory curve here in purple, noticing it goes up and becomes more abnormal over time. And here the sort of clinical function, if you will, activities of daily living, et cetera. So what Cummings et al. Say in a different paper is improved mechanistic understanding of disease onset and progression is central to more efficient AD drug development and will lead to improved therapeutic targets, therapeutic approaches to targets, and we hope so. We hope that, in fact, we'll be able to go all the way back here, diagnose disease very, very early, and be able to institute therapy before the disease progresses. Now, confronted with trials that are failing and with uncertainty as to even how best to treat and when to treat, it's important to note that there are other risk factors for dementia that are quite powerful and modifiable. There are risk factors listed here which can be changed, and they account for almost 40% of risk of dementia in the general population. So a very nice recent paper by Livingston et al. In Lancet talks about these factors and speaks to how powerful they are, and they make several points. The first is that one is in a position to intervene across the lifespan to prevent dementia. It's never too early or too late to intervene. And they make note of the fact that risk factors and interventions do differ in their impact. So here's the list of risk factors, and they're played out in a very powerful way with respect to recommendations. So the commission makes these recommendations. One should minimize dementia, or diabetes rather. One should treat hypertension. One should prevent head injury, stop smoking, reduce air pollution, one of the most recent new factors added to the list. Air pollution increases the risk of dementia. One should reduce midlife obesity, maintain frequent exercise, reduce occurrence of depression, avoid excessive alcohol, treat hearing impairment, maintain social contact, and encourage education at all phases of life. And the idea here is that by doing so, one will have reduced neuropathological damage and increased cognitive reserve and resilience to prevent dementia. Now, when we're looking at these risk factors, these 12, it's important to say that they're acting at different points in life. They're most powerful at different points in life. So less education would be an early life intervention, something that we should seriously consider as being important for our world, where some children have very low levels of education in underdeveloped countries. And in midlife, hearing loss and traumatic brain injury and hypertension and excessive alcohol use and obesity, they intervene to increase the risk of dementia. And in late life, smoking and depression, social isolation, physical inactivity, air pollution, and diabetes. Together, these account for 40% of the risk of Alzheimer's disease, modifiable risk of 40%, unknown risk another 60%. But think about it, ability to reduce one's risk of Alzheimer's disease, of dementia, by 40% by taking these simple lifestyle interventions very seriously. Now, when one looks at the individual factors, one can talk about a relative risk. So, for example, the relative risk of dementia with lesser education is 1.6. So 1.6 over 1, so a 60% increase in the risk. And there's a confidence interval here, but 1.6 is pretty significant. Hearing loss, 1.9. That means that people with hearing loss have a 1.9 times chance of having dementia. Hearing loss is modifiable. And we go down the list, injury to brain, hypertension, etc. What has the last sort of column here, one takes the increase in risk and one multiplies it by the part of the fraction, the fraction of the population affected, to come up with a weighted population adjustable fraction. And that's where we add up all these factors and all the percentages, and we come up at the bottom with the 39.7%. That's where the 40% modifiable risk comes in. Quite striking and quite sensible and quite powerful. And in the same article, the authors talk about COVID-19, and they argue that illness and distancing might increase carer and elder stress and loneliness and neuropsychiatric symptoms and use of psychotropic medications, and thereby further enhance dementia symptoms. And notice here, if you will, that if you take this last column, depression and social isolation are together almost 8% modifiable risk factors. In other words, if one could address depression and social isolation, one could perhaps reduce the risk of dementia by 8%. And that's quite powerful, because notice that other factors may be more powerful, but together, these are really quite influential. So what did we think about this? And we thought we should do something about this. And by we, I mean my colleagues and I in the Institute for Empathy and Compassion at UC San Diego. This was an institute made possible by a gift from Denny Sanford, who you may know. It was established in 2019, so really just last year, to explore the brain bases for empathy and compassion and create training programs to enhance them. We have centers focused on basic and applied research, on medical student education, on faculty and student wellness, and on compassionate patient communication, all things that we view as helping doctors and healthcare givers be more effective with respect to patient care. And we're very eager to engage our community more generally and to support collaborative research on empathy and compassion. Well, when COVID-19 came along, we thought we should do what we could to reduce stress and increase well-being and reduce social isolation in the community at large. And what came to us was, well, maybe we could collaborate with the Center for Mindfulness in providing free online facilitated meditations for mindfulness and compassion. And we did it, and we got a very good response. And we said, well, look, let's not just do it because we think it's helping. Let's prove that it happens and it helps. And let's make it more broadly available. This is an effort led by Dr. Fadal Zidan, who is in the Center for Mindfulness and the Compassion Institute. We collaborated with both the Center for Mindfulness and the Compassion Institute in putting together a program. We provided sessions. Attendees were asked to volunteer for an anonymous brief survey taken before and after each session. The sessions themselves were about 30 minutes, and they featured exercises to enhance mindfulness or compassion or both. Volunteers could choose one or more than one session. They attended whenever they wished. We had attendees that were on average age 50, but fully 22% were over age 65. Volunteers used the visual analog scales to record how they were experiencing stress and anxiety and depression, and to what extent that they were socially connected. The visual analog scale asks you to sort of move a pointer more to the left or more to the right. More to the left is less, more to the right is more with respect to all these measures. We collected data until we reached 200 participants. So 200 attended one session. Of those, 52 attended two and 30 attended three. We analyzed data using a standard package. And the results that I'm going to show you report the change in subjective assessments pre versus post sessions or between sessions. So let's look at the data. First point is that meditation reduces stress after brief mental training. After an individual session of about 30 minutes, we're seeing a decrease in stress of about 60%. That's in session one, with equivalent changes after sessions 2 and 3. All these were statistically significant. And meditation reduces depression in the same way, a little less powerful, but still quite significant. 30 minutes of mental training reduces depression and increases social connectivity. So a pretty nice increase after session 1, but a spectacular increase after session 2. And again, a lesser increase, but still very significant after session 3. Then the next question became, but does this help the elderly? Are people over 65, like myself, benefited by this? So here are the data for those over 65. Yes, we're decreasing stress quite significantly, at least to the same extent, after 1 or 2 or 3 sessions. We're reducing depression even more powerfully than the total group in those over 65 after 1 or 2 or 3 sessions. And we're increasing social connectivity a little bit after session 1, but a tremendous amount after session 2. And again, a goodly amount after session 3. And again, all these changes were statistically significant. I asked Dr. Zidane then to plot the data so that I could understand whether what happened in session 1 carried over to session 2. So here he compared the score before session 1 with the score before session 2. And one can see that even after a delay of roughly 5 days, on average anyway, there were decreases in anxiety and depression and stress, and a small increase in social connectivity, suggesting that one is going to a point with volunteers where these small changes, however real, become more part of their being and less kind of responsive to an individual session. In other words, they're going from state-like changes to trait-like changes in well-being. Very, very helpful and very encouraging. And so we've said that we now need to do a randomized control trial. We have a proposal from Dr. Zidane that we're reviewing right now, and we're going to run this out very, very soon. We'll hope you'll be willing to take part in this. We want to know as best we can, in a very rigorous way, what kinds of sessions work, how well do they work, how often do they need to be given, and what kinds of changes occur, not just subjective changes, but in those around the person who undergoes these sessions. What other changes do we see that are more objective and that translate to those who work with individuals who are actually engaging in such sessions? So I'm going to conclude now by saying that AD is an oncoming epidemic that will require innovations in both molecular treatments and lifestyle interventions. Brief meditations presented by Zoom resulted in significant decreases in anxiety and stress and depression and increased social connectedness. The effects definitely extended those over age 65. The changes were somewhat durable between sessions, separated by an average of 5 days. As mentioned, we'll be undergoing a randomized control study using standardized measures to evaluate specific practices in brief sessions given over 7 days. And of course, we're eager for you and for others to join this in future efforts aimed at enhancing well-being of elders and their caregivers. And with that, I'll stop and thank you for your attention.
Thank you, Dr. Mobley. If anybody has any questions, please feel free to use the chat function. We do have one question, and that is, we have several, but let me get to that. So this question is from Fran P. She asks, is there an age that a person should seek testing for preventative purposes?
Right. I think that the recommendation now would be that one should, over the age of 60, this is a recommendation. I don't think it's written down anywhere, but my own advice would be that over the age of 60, one should, at their yearly visit with their physician, undergo brief screening and screen testing for memory concerns. Excuse me, I can say after attending our memory disorders clinic, from the physician perspective, it goes very, very quickly. The tests are relatively simple. They're not stressful. They're kind of fun in a way. So I think that over age 60, it's quite reasonable to have yearly testing for memory concerns. If the screening test is abnormal, then we have a protocol that has people going on to more specific testing to more fully evaluate whether there's a problem. And then, of course, if those warning signs are there and further testing shows some concern, then one can go on to further, more specific testing along the lines of the biomarkers that we've talked about.
Thank you. Denise C. asks, when you said that psychotropic drugs contribute to dementia, isn't that a treatment for depression?
Yeah, it is, of course, and it's true that depression is treated frequently by, for example, serotonin release inhibitors, serotonin uptake inhibitors, so that serotonin levels are higher. They're psychotropic. Those for depression could obviously cause problems with cognition. The dose of those drugs, the drug combination used in individuals is very important. And so one would ask their physician, you know I'm taking these drugs, is it likely to compromise my cognition? I think what one's concerned about here are doses that are very high or combinations of drugs that could compromise one's ability to remember and learn and perform cognitively. But it's a good question, and certainly one should ask their physician, are you concerned that these drugs that I'm taking and the doses that I'm taking could compromise my function?
Great, thank you. Janet V. asks, how can one participate in the next study?
Yeah, so I've given Nancy our web link, and we'll be announcing that trial on our website. You're very welcome to volunteer to be part of that. What will happen is I think we're looking for 280 people to be part of that trial. People will volunteer to participate. There will be very few people that will be ineligible. Your willingness to volunteer is probably the only real criterion that would be used. And then you'll be randomized to one or another group. Some groups will be active treatment, other groups will be controls, and you won't know what group you're in. And then after this brief testing, brief set of sessions, you'll be tested throughout, and at the end of that time you'll be tested. And then once we have completed the study, we'll break the code and you'll know what group that you were enrolled in. And then I think from that, what we'll say is, look, maybe session type 1 and type 2 were the most effective. Now we'll offer those online again for free so that people can take advantage of those sessions and enroll in them on a more continuing basis. So we want to get to the answer, what kinds of practices, what the dose is, how often they should be taken before we start offering them, because we don't really yet know what's the most effective. But yes, if you go to our website, you can volunteer, you can be enrolled, and eventually you'll understand what group you were in and what recommendations we have for future sessions for you.
Perfect. And I'll put up that website in just 1 minute, a little bit towards the end, we'll get through the questions. Glenn R asks, when or how is the amyloid produced? How is its presence detected? Is there any connection between amyloid cardiopathy and Alzheimer's?
Yeah, so amyloid is this sticky substance that results from the production of the A beta peptide. Those peptides bind to one another and make up this kind of gel-like mess, a gel-like substance in the brain. That's what is present in plaques. It's important to say that amyloid plaque is only one manifestation of the molecular nature of those A beta peptides. They also exist in small assemblies that are soluble, not really gel-like, and that they can injure neurons. But the answer is it's a product of the amyloid precursor protein. The cells that make the amyloid precursor proteins are mostly neurons. They're detected clinically through PET scanning, amyloid PET scanning. And the amyloid that's present in the brain is different than the cardiac amyloid. So there's a bunch of different kinds of peptides that produce an amyloid-like state. This peptide is not the cardiac amyloid that one thinks of as cardiac amyloidosis.
Thank you. Barbara B asks, if your parents or grandparents had dementia, would you recommend an MRI or other testing without symptoms at a certain age?
So ask the question again, Nancy, sorry.
If your parents or grandparents had dementia, would you recommend MRI or other testing without symptoms at a certain age?
No, I wouldn't. I would, Alzheimer's disease, again, is really quite common. By age 85, for example, maybe 40% of people have Alzheimer's disease. So we have to contend with the fact that our elders getting Alzheimer's disease was not a particularly surprising or a unique event. It was almost the expectation if they were quite old. It wouldn't be a good use of resources to start scanning at a specific age unless when we're in a specific clinical trial looking at the natural history of Alzheimer's disease. Rather, I think one would simply do this regular memory testing at the physician's office beginning at age 60 and avoiding any further testing unless there were warning signs shown by those screening tests and follow-up tests.
A popular question is, what does less than 21 units of alcohol per week translate to?
It's a lot of alcohol. The answer is it's a lot of alcohol. I think they mean the equivalent of 21 shots of alcohol, which should probably be, I guess, 3 shots a day. And, that's a lot of alcohol. And I mean, the message here is please avoid that. It's moderate alcohol. I mean, cocktail a day or a glass of wine a day. I mean, there's evidence to suggest that that's actually good for cognition, good for memory. But we're looking at 21 shots a week that's much more than, one glass of wine a day or even 2 glasses of wine a day.
Thank you. Barbara S. asks, is there any marker that could show up in our annual blood work? Can we specifically ask our doctor to include that in our lab results?
Not today. I'm thinking the reference would be to just blood or plasma. There are tests coming along. There are tests that exist right now that will show a pattern consistent with dementia in those at risk for dementia. I don't think we're at the point where those are being used now broadly. I think one would only use those tests where there's some concern for memory loss. Now, having said that, we're entering a new era in which very early studies to diagnose changes as referenced in plasma in A beta 42 or in p-tau are coming available. And it may well be in just a few years that absolutely normal 60-year-olds will be getting such testing to anticipate, right? To anticipate the later onset of dementia. That I should have pointed out in that one graph where we showed, abnormality of a biomarker versus age. Alzheimer's disease pathogenesis probably begins a couple of decades before one sees memory loss. And so increasingly, we're focusing on that early period of time when there's no memory loss. and asking which biomarkers work. And I think right now one can say with some confidence that we will have blood tests that will be taken routinely in the population as a whole. In other words, outside of a clinical study in years coming forward. And there'll probably be tests for A beta 42, for certain phospho-tau species, p-tau species, and likely for this protein called NFL. But we're not there yet. So as yet, I think we just have to wait on that to see when that generally becomes available for us.
Thank you, Dr. Mobley. There's another question from Faye G. She says, how closely are the clinical signs and the physical/brain? I'm not sure if that's clear enough for you to.
I think what she's referring to is the thing we just talked about. You know, if I were to ask the question, which comes earliest? Is it the memory loss or the brain changes or these subtle changes in biochemistry and synapses? It's the latter, right? So we're seeing changes in synapses and biochemical markers a decade before we're seeing significant brain pathology and memory loss. So I think that's what she's saying. It's fair to say that where you see brain pathology, you're likely to see clinical symptoms. But these subtle changes in the biochemistry and these subtle changes in synapses predate almost by 2 decades any real change in significant brain pathology or significant cognitive decline.
Thank you, Dr. Mobley. I'm going to go ahead and share this screen so that we can remind everybody about the website where they can find the mindful classes.
People are encouraged to come, and it's really fun, and we have wonderful teachers, and we hope to see you online, and we hope that we'll continue this discussion and collaboration, and that what we do will actually benefit you.
Wonderful. We'll also share this information on our website so that it's an easy resource for you to access if it's something that you haven't written down yet. A big thank you to Dr. Mobley for sharing all his research and insight on this very important topic. As a reminder, at the Community Center, we also have a lot of wonderful programs that we encourage you to join us for. We have daily classes, including meditation, Tai Chi, yoga. Not only are these wonderful programs for you physically and mentally, but it also allows for some socialization, which is so important, especially during this time. So just a reminder, we have our Wednesday Connect series. It's every Wednesday we have a wonderful speaker, and it's a very interactive program that we hope you all can join us for. We have, again, free virtual classes on our website. That's ljcommunitycenter.org, and we'll also link Dr. Mobley's the Mindfulness Institution classes there too. One more thing, we have a virtual mural candidate forum coming up on October 22nd. That's also a free program, and we really hope that you can join us for that. And we'll end here with the Sanford Institute. Dr. Mobley, thank you so much for joining us today. We really enjoyed having you join our Distinguished Speaker Series and enlightening us. And if anyone has any additional questions, please feel free to email us. We'll get those questions over to Dr. Mobley. Thank you all for joining.
Thanks so much. My pleasure. Take care.
Thank you. Stay well.